All right, so this is a depiction of the C4 cycle. So just to be clear, all plants on the face of the earth, they do the C3 cycle. It's just that some plants have evolved these additional pathways that they kind of tack on top of the C3, so to speak. And so they're linked to the C3. But all plants can do the C3. And then C4 plants, they have the C4 cycle where you can see that they don't have rubisco immediately work with that CO2. They have that PEP carboxylase doing the job. So the CO2 comes into the plant through the stomata. The PEP carboxylase enzyme is there to react CO2 with PEP, phosphoenolpyruvate, and then you get oxaloacetate, which quickly converts to malate. Malate then is the vector that moves over to the bundle sheath cell because it's got to get over there because that's where the Calvin cycle is, right? You don't see the Calvin cycle running in the mesophyll. It's in the bundle sheath. And so the Calvin cycle is tucked away because in the mesophyll, you have the light reactions going on. And remember, the light reactions evolve off oxygen, and rubisco that's in the Calvin cycle, you don't want oxygen in that bit. All right, so these plants have tucked away their rubisco into the bundle sheath where there aren't light reactions cooking along. So that means there's not a lot of oxygen in the bundle sheath. So that's great. And then they have malate do this job of, pretty much flooding the area around Bisco with lots and lots of phytoplankton. And as long as you keep this cycle going, that will work. You can see that once CO2 is delivered by malate, essentially CO2 is part of the malate molecule, and then it gets cleaved off and given to rubisco, and then rubisco reacts that with RGP, and then the Calvin cycle comes along. So then we got to keep the C4 cycle going though. So that's where you see the pyruvate is the other product then. And after the CO2 is delivered, you end up with pyruvate, which has to get back over to the mesophyll. And you can see it has to get a little bit of energy applied to this. So ATP is going to be used to convert pyruvate to phosphoenolpyruvate. Got to get a phosphate group on there. And so there's an extra cost to running this cycle, but apparently it's worth it because the benefits outweigh the cost. C4 plants, they've evolved this mechanism and they've maintained it over millions of years. So then once you get that phosphoenolpyruvate regenerated, then it's good to start all over again. The C4 cycle coming in, the CO2 coming in and reacting. Questions on the C4? Yes. Yeah. Yep. Yep. With C3 plants, they don't have any like front runner enzyme. When CO2 comes in, it's rubisco that it will react with. Other questions on the C4? Yes. So yeah, I mean, typical leaf cells, they refer to the inner leaf cells as mesophyll cells. That's just they're just parenchyma cells. When people say mesophyll, think parenchyma. That's the kind of cell they are. They just had to give them a special name. When you hear mesophyll, think about leaves, and that's what's inside leaves. So, but they're just parenchyma cells inside leaves. Just they scientists love jargon, unfortunately. This is, I think, from your book. It's saying the same thing. So whichever one you want to look at to learn off of, that's fine. So I think we were last time looking at this idea of the internal anatomy of the leaf is a little different for C4 plants because their bundle sheath cells are so important and they're running the Calvin cycle exclusively. They really clump up with a lot of chloroplasts. They're just bigger, much more visually striking when you look at them under the microscope. And then the surrounding mesophyll cells tend to kind of hug up to the bundle sheath. There's lots of plasmodesmata connections, so you can have that flow of malate and then pyruvate going round and round and round so that everything is cooking along correctly. The artist tried to do that here, although I wish they would have been a little bit more generous with the pumping up of the bundle sheath. But they're nicely showing how the mesophyll cells are hugging the bundle sheath and allowing for that nice flow of molecules between the two. And then I think this is where we left off, looking at the C4 plants examples. And so I've updated this slide set, by the way, to cover, so this is new, it's in EduCAD, it's updated. And so C4 plants, there's about 19 different families where we could find C4 plants. The grasses are really popular. They have developed a strategy, and if you think about it, a lot of grasses are native to tropical and subtropical areas. And so those are types of environmental conditions where the C4 cycle makes a ton of sense, because you have consistently hot conditions and then droughty periods. And so it makes sense that C4 cycle, what it allows these plants to do in that kind of environment is they can close down their stomata really, like just barely open, just kind of sip in CO2, and they can still be very, very efficient at making sure Rubisco just sees CO2. And then the photorespiration gets suppressed. Right? About 3% of all known species, so not a huge number, can do C4. Some very important crop plants, of course, but also some weeds that might pop up in your lawn. And that's why weeds tend to sometimes outcompete some of your native grasses under drought conditions in particular. They see that the C4 pathway has really been independently evolved. So there's been a lot of trial and error from many different plants. So it's been happening independently over time. So it's not like one plant figured it out and everything stems from that in terms of the C4. There's been a lot of attempts by a lot of different plants. So 40% greater ability to convert sunshine into biomass than just straight C3 plants. Maximize growth rate and yields, 40 to 50% greater than those of the most productive C3 plants. So again, this does give them a nice edge, especially under consistently hot periods and droughty periods. Okay? So that's why they're looking at making the rice plant to be C4. Rice plants are just C3, but they're trying to genetically modify them to run the C4 cycle. And they're making some progress, but it's slow going. That's a lot of different enzymes and gene expressions and gene modifications that need to be done, but they're working on it. Okay, so C4. The other pathway I need to talk to you about is called the CAM pathway. And the plants that do it are called CAM plants. Again, when you look at what's happening, it is a mechanism to concentrate CO2 around here. So that Rubisco really doesn't see much oxygen at all. CAM, I have it written in one of these slides. CAM stands for crassulacean acid metabolism, which is a mouthful. So we shorten it up to CAM. The C that stands for Crassulaceae, that's a family of plants, and it's a lot of succulents are in that family. That's where it was first discovered. Unfortunately, since then, we now know that there's other families of plants that do it, but this name had already stuck. The acronym was nice, so they just kept it. But I'll explain who else can do the CAM in a bit here. Now, luckily, what we see happening in this pathway is pretty similar to the C4 in terms of some of the same exact players. PEP carboxylase is the first enzyme that works with CO2. So as the succulents or any of the CAM plants are bringing in CO2, it's going to work first with PEP carboxylase, just like what we saw in the C4. And then it reacts it with PEP, just like before, C4. And then you end up with oxaloacetate. just like before. So let me show you, this one's a little bit more detailed in the next slide. Let me go to this one. So now what we see is the artist is trying to show that something is happening during the day and something is happening at night. So we said C4 plants have spatial segregation, right? Some things are happening in mesophyll cells, other things happening in the bundle shape. But it's during the day, all day long. Cam plants, they're really unique because they can do some things at night and then during the day do another aspect. So think about it. If you are a giant saguaro cactus and you're living in New Mexico, and if you've ever visited that area of the United States, would you want to open your stomata at high noon? No. You know, it cracks over 100 degrees regularly, 100 degrees Fahrenheit regularly there. Sunshine all the time. I once had an advisee who moved out to Arizona to do her PhD out there, and she came back to visit. She's a native of Escanaba. She went to Northern, got her bachelor's here, then she went out west for her PhD, and she came back to visit one summer, and I'm like, oh, Jenny, how do you like Arizona? You know, so different from here. And she says, I don't really like it. I'm like, what? And she said, there's no clouds in the sky. It drives me nuts. So here we're used to a lot of beautiful clouds. I mean, that is one thing I really love about this area. Nice, poofy, beautiful clouds. And she said, I can't stand it. There's no clouds, just sunshine all the time. And so I had to laugh at them. I think I got it, but it was interesting to hear. So again, if you're giant saguaro or cactus, opening your stomata, no clouds to shade you, nothing. And it's over 100 degrees frequently and drought, long drought periods. So yeah, no, that doesn't make much sense to open your stomata. You're going to dehydrate and die pretty quickly. So they don't. They keep their stomata closed. But they got to run, you know, photosynthesis. So how are they going to do this? Well, during the night, that's when they open their stomata. At nighttime, the sun goes down, humidity comes up a little bit, temperatures come down. It's more conducive to having your stomata open. So go ahead and do that. So here on the bottom of the image, there's CO2 coming in at night and reacting with PEP with the help of the PEP carboxylase enzyme. And then you get oxaloacetate, which then gets converted to malate. So very similar to the C4. Except now what they do is, because it's nighttime, you can't run the Calvin cycle at night because some of the Calvin cycle enzymes need light to be fully active. So there's a conundrum. It's like, okay, we can do the first part, but then we got to store up our malate. Essentially, that's, you know, malate has the CO2 there. It's part of it. So they go, well, okay, let's just put it into the vacuole. We'll just stuff it into the vacuole all night long. And that's what they do. And so their vacuole gets stuffed full of malate during the night. And then as the sun comes up and strikes the giant saguaro, it makes sense to go ahead and close down your stomata. But now that the sun is striking the plant body, they can kick into photosynthesis with the light reactions of the Calvin cycle. And so what they do during the day is they mobilize the malate out of the vacuole over to the chloroplast in the same cell. We're not shifting cells. We're all working within the mesophyll cell. All right, so CO2 comes out of the vacuole, goes into the chloroplast of the same cell, and will be right where, you know, Rubisco's sitting right there, waiting to get that carbon dioxide and react it with REBC. So they have this day-night rhythm of doing certain things during the night when it makes sense, so they don't die of desiccation. And then in the day, they run light reactions and the Calvin cycle. So really a very good adaptation. So we're thinking about plants that live in extreme heat conditions, all right? So the desert environment, where it's extreme heat, long periods of drought. Yes, they do get those monsoons that sweep through the Northwest area, but in general, they have long drought periods. Okay. So this is in words, what we were just talking about. Okay. So those organic acids, that's the malate, malic acid. Malic acid is acid, and it's just filling up the vacuole. If you put a pH electrode into the vacuole all night long, you can see the pH getting more and more acidic, dropping and getting more and more acidic. And then during the day, they just mobilize the malate out, have it as a vector to go from the vacuole to the chloroplast in the same cell, so it's less distance to travel compared to C4. But again, malate is delivering the CO2. So that Rubisco is getting bathed with CO2. Yes, malic acid. Anytime you see ATE on the end of a word in science, that means it's an acid. So oxaloacetic acid, you can say oxaloacetate, and that's the same thing. Yeah, oxaloacetate is the same thing. Malate, so same thing. Just depends on who's talking, how they prefer to say it. Questions on the cam? Yes. Well, let me show you. Let's see. Do I have it in here? Well, yeah, let me scroll back to this one. I won't get into the details when I teach plant phys, but basically what we have is, in order to get PEP available, CAM plants actually have starch stored up in their chloroplast. And then when they're like, hey, we need some PEP, they'll please starch, break it down, and the reaction happens. There is some level of ATP, so there is a cost. I can't remember the numbers per gram, but there, yeah, there's a starch. And so, but they break down starch in order to have a source to build their phospholipid. So again, it's, you know, there's these extra pathways, but when you put it into context of where these plants are native to, where they evolved to live, it does make sense that they have these extra pathways in order to deal with some very challenging environments, you know, where opening your stomata could be deadly. And so, but yet they need the CO2. And so it's really you know, a tight little situation where you have to be very, very careful of, well, should I open my stomata or not? And how much should I open my stomata? And they're constantly assessing their availability of soil water. Like, how much can I open my stomata before I start going into water stress? And so they're being rooted in place and not being able to go to the refrigerator and get a glass of water can have a huge impact on these plants. So like I said, there are, you know, plants outside of the family Crassulaceae that can do CAM. But if you look at a lot of succulents which are in the family Crassulaceae, yes, they are, but not every single one does. It just depends on which one you're looking at. And so the other thing, pineapple is a huge commercial crop. On this picture of these images, pineapple is big business. And then there's some like prickly pear and some like that are economically important. This plant up here in the top left, that's Mother of Thousands is the nickname for that one. It's a calancho, but it has little baby plantlets, clones of itself. It's little clones of the mother plant, little leaves, the roots dangle off, and then you know, something brushes against that, those little plantlets will drop off. We used to have a bunch of those in the greenhouse, and sometimes if people used a watering hose to spray the plants, you know, the hose off the foliage, those would break off, scatter into pots all over the place. Pretty soon we had calancho growing in all these pots. It was sometimes a little bit annoying how much that plant is fun, but it can grow. The giant saguaro that I mentioned, beautiful, beautiful, amazing plant, state flower of Arizona, if any of you are from Arizona. So, it's my understanding they're protected. You cannot, like, if you have a giant saguaro on your property, you can't like say, oh, I want to cut it down. I don't like it. You have to get special permission. I think they actually like move it. If you really want it off your property, they'll like move it and try to keep it alive. So anyways, so let's look at CAM. We have the family Constellaceae, but there's like 38 different families that you can find a CAM plant in those. So, and not every single species in that one family will do CAM. Some families do all. You'll see C3 plants, CAM, C4. They think about 60 different times this CAM pathway has evolved over time. Vanilla, the vanilla orchid, that's the only orchid we get something commercial from in terms of something to eat, so to speak. So the flavoring, the vanilla flavoring. Orchids are big business in and of themselves, however. I mean, that's $1 billion industry right there. Pineapple I already mentioned. What do we get? Agave. What's tequila? Yeah. I have fond memories of that. So, grad school, lots of fun. So, but more primitive plants too. So let me go back to this picture. This lower left picture is a more primitive plant than the angiosperms. That's actually a gymnosperm, believe it or not. Does not look like a typical gymnosperm, okay? And we'll learn more about that plant when we get into the gymnosperms, but that lower left one, that's called Welwitschia, and it's a bizarre plant that, as far as I know, it only exists in Namibia, and only gets its water from fog, pretty much fog coming off the ocean when it gets foggy. So really interesting plant. So they can find the CAM pathway in more primitive plants than the angiosperms. So what that is implying is that you have the evolutionary process, the lower bullet point. So the first thing to evolve was the C3 cycle, and actually bacteria came up with the C3 cycle, not plants. Right? Bacteria did it first. Then plants came along and said, yeah, okay, I like that, I'll do that. And then the CAM cycle or pathway came next. That was the next invented thing through the evolutionary process. And then the C4. So definite process that they see when they look at the evolutionary process of these pathways. The CAM pathway is actually gaining momentum in terms of, hey, maybe we could make use of the CAM pathway in the sense of, like I said, they're trying to make rice plants C4 plants. Well, they're looking a little bit more closely now at CAM. And they're saying with global climate change that we need to think about this idea and ways to solve it. And they say that they're increasingly recognized as among the world's most important climate resilient crops for food, forage, fodder, fiber, and fuel. And so I've been seeing more and more published articles on this idea, where what can we do if we can better understand the CAM pathway? Can we genetically modify plants or somehow create these other plants that have properties similar to these CAM? And so that's starting to be a thing that they're working on as well. It's not just the rice initiative where they're trying to make the rice to be a C4. So we're thinking about environments like this, the beautiful Southwest, where you have those really long, dry, hot days. Under these different cycles, going back to this, you can see the water comparison. So the C3 plant, not so good. They lose 400 to 500 grams of water per, when they run the C3 cycle. But you can see how much better the CAM is, either C4 or C3. So very, very good at water conservation. You can shut your stomata all day long. And especially in those high heat conditions, that's a really good strategy to conserve water. And now you know why a lot of plants in the desert are often, very spiky and dangerous to touch or to try to eat. because they are good at conserving water. They have water stored up, where a lot of other animals or plants that can't do the CAM, not so much. And so they have to defend themselves against all the things that want to come along and not only eat their tissue, but get to their water as well. So, all right. So now I've been talking a lot about the C3 cycle, the C4 that's tacked on it, the CAM, that's the ignite process. So what about sucrose? We haven't really, I've alluded to that. So when you run the Calvin cycle or the C3 cycle, there's a point in the cycle where once that molecule gets made to a high enough level, then you can put it into sucrose. You remember what the three-letter acronym is in the Calvin cycle? Get it built up, and then some of it goes off into sucrose. What's the name of that molecule? G3P. And that's the other thing too. I've seen in the past students confuse PGA and G3P. And it's easy to do if you haven't spent some time. So make sure that you understand the Calvin cycle process and that it's the G3P that will, once enough of that builds up, some of it gets siphoned off sucrose. So that's what you're seeing here. And I'm not going to go into detail on this. I covered it a little bit more in that big, but basically just to show a visual, Calvin cycle is cooking along in the chloroplast. Once the triose phosphate is the G3P. Okay, so once the triose phosphate or the G3P gets made, then if there's enough of it made, some of it gets put out into the cytoplasm or the cytosol. And then it goes into this pathway where you can see finally at the end, sucrose is made. Okay, so that's why the plant wants to run the Calvin cycle, is to get enough G3P building up so it can siphon some of that off into this sucrose synthesis pathway, right? It's tightly regulated. There's a special transmembrane protein here that is tightly regulating this process because it's a delicate balance between I got to keep the Calvin cycle running all day long, but I got to get my sucrose made too. And so it makes sense that there's quite a bit of regulation on it. Okay. So yes, sucrose is getting made. That's the sugar the plant cares about, not glucose. It wants to make sucrose. So another reason I put this in here is to visually show you they're shooting to make sucrose. All right. And so many other people, they think it's glucose if they haven't taken a botany class. Okay. And then this is just to show the idea of, how do these plants react? These are C3 versus C4 under different environmental conditions. And so on the left graph, you see light intensity, essentially. Light absorbed versus rate of photosynthesis on the y-axis. So higher light intensity, you can see that the C3 plants plateau, they level off. So that means that, once they reach 400 they start to plateau. However, C4 plants keep more. And so you give them more light, they do more photosynthesis. More light, more photosynthesis. More light, more photosynthesis. And you can see they really barely plateau all the way out, getting close to 1,000 there. So they're just better at absorbing that high light. They're good in high light. That's when you look at their tropical, subtropical native habitats, they're used to it. They're used to very high light. And so they outperform the C3 plant in terms of rates of photosynthesis. Temperature, think about climate change where some areas of the world are going to get consistently warmer. You can see again, C3 plants, they're a little bit more sensitive. They start to tank around 30 degrees Celsius. C4 plants are like, yeah, I'm used to this. This is how I evolved. I'm used to hot temperatures. I can keep going. My rates of photosynthesis are still good, all the way to over 40 degrees. So they got an edge there. And then here on the last one, CO2 concentration. You've been learning how with global climate change, all of that, CO2 levels are increasing. And so the question is, well, plants love CO2. Isn't that a good thing? And it's like, well, yes and no. You can see that C4 plants, they're so good at taking in CO2 and being so efficient with it that they actually plateau quite early. More CO2 to them isn't helping. They're already really good at it. But with C3 plants, you can see if you give them extra CO2, they like, oh, okay, yeah, because that starts to minimize the PCO cycle. And so remember, C3 plants are pretty, it's pretty risky. The PCO cycle can run under the right conditions pretty easily in the C3 plant. So if you increase CO2 around the C3 plant, that's good. They can continue to do better rates of photosynthesis. C4 plants are already so good at it that they don't really, it doesn't matter. So it's complicated. Yes, more CO2 in our atmosphere, it could help C3 plants, but it really doesn't do much for the C4. They're already very efficient. Okay. Questions on any of the CAM or sucrose synthesis or anything? Yes.
Lecture, but I was wondering if we're going to be able to review the exam multiple choice.
Yes. Oh, thank you for reminding me. So today's Friday. I'm behind because I was sick earlier. I am hoping by Monday's lecture, I'm going to go over some of the exam with you all in lecture. And then if you want more detail than that, then setting up an appointment with me. Yeah, then I actually would prefer if you want to see details that we meet one-on-one, because then I can tailor how to help you the best rather than if there's a bunch of, you know what I mean? So, yeah, send me an email or talk to me and we can set up an appointment. So, it's good to do that before the next exam, right? Because the next exam is coming up in a little while. We have 2 weeks out, I guess, is the next exam. All right. Well, there's 4 minutes left. So what I'm going to do, like I said, I'm going to talk to you a little bit about the exam on Monday after I need to analyze the stats on it first. So I want to do that first so I can see where there were some stumbling blocks, I guess, and go over that with all of you. And then on Monday also, I will get into more of the roots. So you looked at roots yesterday in lab. So I'll wrap up the roots lecture on Monday. There's a few more things I want to say. And then on the syllabus, it's talking in week 4, we were supposed to cover water in soil, and I didn't get to that yet. We will. It's just going to get interspersed a little bit because when I'm talking about water, I'm mainly focusing on how water moves through the plant body. So we'll talk a little bit about how it gets into the plant and then how it starts to go through the xylem. So you will be getting that content. It's just a little bit later. Okay, so we'll start roots on Monday, or finish roots, I guess.